So next we're talking about structures and functions. So structures, organisms have developed many different ways of doing things that keep them alive. The structures or body parts they have developed to perform these tasks have different functions. So insects have developed spiracles, which are holes on the sides of the abdomen in some insects. The insect can adjust the size of the spiracle to control the amount of air entering the body. This is their respiratory system. This is different than mammals that we use our lungs, right? So this is something completely different. And we have functions. So all organisms have built-in structures that perform certain tasks or functions to stay alive. And these tasks may include gathering and processing nourishment, generating and exchanging waste, responding to the internal and external stimuli, adaptations, protections, reproductions, movement, locomotion, which is something that's very important. Okay. So different structures can have similar functions. So different plants and animals have developed different structures for doing similar functions. So a function of food gathering, a plant uses its roots, whereas animals may use their claws, hands, tentacles, mouths, and tongues. Okay? For the function of breathing, a plant uses its leaves and needles, and it breathes in carbon dioxide, whereas animals use their gills, lungs, spiracles, and skin to breathe in oxygen. Okay? For the function of moving, we know that plants don't usually move from place to place, but they move in one place, right? They do this stationary dance of sorts. Whereas animals have wings, legs, fins, tails. We use kind of lots of different things, right? This is the idea that the plant is more stiff than the animal is. Okay? Similarity in structure with some variability can be seen among animals living in the Galapagos Islands. Charles Darwin studied many of the animals in the Galapagos Islands and determined the origin of species as a result of his observations. So, from that, Darwin, he looked at closely related species of finches that have different bill structures to perform the function of gathering food. So he called this adaptive radiation. So where you have one type of finch and he kind of goes off and the rest of these finches have adaptations. Okay, so you have the original finch that eats seeds. Okay, and then you have a different sort of beak for eating buds and fruit, and a different one for eating grubs. And then there's a finch that uses a tool to get its food. There's a specific type of beak for insects or leaves. Okay, so it's this idea that you have one central one, and then adaptive radiation. So these adaptions come out of this one particular original finch. Okay, I'm going to quickly talk about microscopes. So, view of cells using a microscope. So when an object is made to appear larger than it actually is, it's said to be magnified. In early microscopes, microorganisms were first discovered by Anton van Leeuwenhoek, and his hobby of grinding lenses led him to eventually make the first simple microscope to study blood samples, pond water, and plaque, so what you scrape from your teeth. Whereas organisms he found that were single-celled he called anemoicals, so small animals. Albert Hooke was also experimenting with microscopes, and he had built to look at different things, such as a tiny piece of cork. And the small holes that were honeycombed were described as little rooms or boxes, and the word cellulae was used to name them from the Latin form of cell. Okay? Next, we're going to talk about such cells.